Good morning, everyone. I wish I could be with you in this Infrafrontier meeting, but I'm here in Puerto Rico attending yet another meeting. But nonetheless, I'll be delighted to share this spot seminar, this short seminar on these ancestral CRISPR-Cas systems that we have characterized. Okay. We have a main problem to solve, which is that most of the applications that we are doing currently with genome editing tools are related to Streptococcus pyogenes or Staphylococcus aureus Cas9 proteins. And these are pathogenic bacteria for which we have antibodies and for which we have T cells. This was recognized and studied by Matthew Porteous, and it was published a couple of years ago. So most people have antibodies and T cells against these proteins. So this is bad news. Therefore, we need to find out other CRISPR-Cas systems. There are many CRISPR-Cas systems, there are many bacteria, there are many archaea. And so this is what people have done, going through different places, Antarctica, the Atacama Desert, to the Yellowstone National Park, to the Everest Summit. And we also have done something on this area. We just published, together with Francis Mojica, a new Cas9 from a lagoon very near Alicante. So these are new CRISPR-Cas systems existing in other bacteria that for which we expect that they will not be behaving as the CRISPR-Cas9 from Streptococcus pyogenes and will not be recognized by our immune system. But we wanted to do something else. We wanted to find ancestral CRISPR-Cas systems. So we wanted to travel not in space, but we wanted to travel in time. So Back in the Future movie, similar type of thing. Okay, we have a problem. The problem is that we have some ideas of what could be the ancestral CRISPR-Cas systems, and these are current CRISPR-Cas9 systems, and Feng Zhang and others have described these proteins. But we are not interested in these CRISPR-Cas that are existing nowadays. We want to characterize CRISPR-Cas systems from many millions of years ago. There is a problem. There is another problem, which is the oldest DNA we just learned before Christmas is just 2 million years old. Remember that Svante Pääbo, the last medicine Nobel Prize, was studying DNA from 10,000 to 400,000 years, which is a lot, but it's nothing you compare to the life history on Earth. So basically, our planet is 4.5 billion years ago. 3.5 billion years ago, the first bacteria appeared. 1.8 billion of years ago, the first eukaryotic cells appear on Earth. 600 million years ago, it was the Cambric explosion. And then from 150 million years ago to 65 million years ago, the last ones, so the dinosaurs, extinguished. Eventually, we managed to obtain the sequences from the ancestral proteins, 3,737, 200,000, and 2,600 million years ago. How we did this? Resurrecting ancestral CRISPR-Cas in collaboration with a paleoenzymologist, which is Raúl Perez-Jimenez at the SIG Nanogune. So basically, this is reconstructing CRISPR-Cas proteins through a maximum likelihood method. So we are collecting sequences from 60 plus bacteria, and these bacteria, we are giving them to a computer, and then we are asking the computer to obtain the ancestral proteins through a timeline. And so we can go back in time between 37 to 2,600 million years ago. So they are surprisingly similar, these structures, even though the primary sequence is different. This is AlphaVol, and most importantly, these new proteins, they are not recognized by the current antibodies. As we are going back in time, they display nicking and double-strand cutting activity, but the nicking activity is preferentially displayed by the oldest, which in addition, they have single-strand DNA and RNA cutting activity, which makes sense because this is what we understand, that the life started on our RNA well, and this is what you would expect of a CRISPR-Cas system. We also recognize that they have a PAM-less cutting activity. The oldest you go, the more pumpless activity have. So that also makes sense because this is reflecting the less complexity that they will be existing back in time. But the most important experiment is displayed here on human cells. And basically, we have asked whether they are acting as genome editing tools. And this is what we verified. So in red is the reference Streptococcus pyogenes, and then following 137 to 100,000 and 2,600 million years ago. And as you can see, there is a remarkable activity in two genes that we have tested in these human cells. So basically, the take-home message is that we are able to resurrect these oldest ancestral CRISPR-Cas proteins, and they work as genome editing tools. So the alleles that they are produced, they are similar, and in different types of experiments. So they are not only able to cut these genes, but they are producing similar type of alleles. So this eventually was published in January in Nature Microbiology. And what we're doing now, we are testing this in vivo, both in mouse and zebrafish embryos. I wanted to thank the collaboration. These are the three labs mainly collaborating for this project. Raúl Perez to the right, in the middle Francis Mojica, and myself. So I thank everyone in my group also for contributing to this project.